It's all coming through nice and clear for everybody. So thanks for joining us today, everybody. We're going to talk about surveying mosquito distribution in BC and Yukon Territory in a changing climate. And I'm going to have Danica jump in here and give us some background to sort of start us off on the right foot, as it were. Danica?
Thank you. Awesome. So yeah, as Dan said, I'm just going to go over some basic background information for you all before he jumps into the really exciting stuff. So first off, this is the general life cycle of the mosquito. So it involves 4 main stages. The first one as eggs before hatching into aquatic larvae, which actually go through 4 instar stages themselves, before morphing into pupae, which are also aquatic, and the most middle stage of the life cycle. They're super quick to get away from predators. And then after a couple of days, they emerge into adult mosquitoes. And this whole life cycle takes about 8 to 10 days to complete in full. And as you may know, the adult mosquitoes that you see here, they love the summertime and the warm weather in BC and the Yukon, but they don't just die off in the winter. Some of them are able to diapause and survive through the cold season to the next year. And the stages in which they're able to do that are indicated by the snowflakes in this graphic here. Some of the most common way is in the egg stage, but there are some species in BC that can diapause as larvae, and they've actually been observed under layers of ice, which is pretty neat, before eventually that melts, and they can move on to their next life stage in the summertime. As for the adults, for the ones that diapause in that stage, for females, they usually mate and then go into a state of torpor, sort of hibernation. And then once the warm season comes around, they exit their place and feed to lay their eggs again, doing that multiple times throughout the warm season. So yeah, that's how they survive our cold season here and thrive in the summer. So just to go over a couple of their common pathogen transmission cycles here, which you may already be familiar with, specifically the urban cycle. So how this works is a mosquito will feed on a human who's been infected already with yellow fever, dengue, malaria, and if the species feeding on them is compatible with the pathogen they're already holding, it'll get sucked up along with the blood. And then as the mosquito digests their blood meal, the pathogen will replicate, eventually exiting the midgut and moving into their salivary glands. And what that means from there is that the next time they go to bite someone, a small amount of saliva will be injected into that person when they bite, along with the pathogen that they contain, potentially making that person pretty sick. And this cycle then just continues and continues. The sylvatic cycle is relatively similar in its mode of transmission, but it's more looking at wildlife. So pictured here is a monkey, but mosquitoes are also known to feed on birds, some amphibians, and cattle as well. So potential disease threat in those communities, and also some crossover between these 2 cycles as well. So for example, a mosquito feeding on some sort of wild animal, picking up a pathogen, and then potentially transmitting it over to a human if it switches over on its host. So lots of crossover there. But besides the public health and pathogen side of mosquitoes, they also have a pretty important ecological role as well. So although females take blood meals, they also need to feed on nectar throughout their life for energy and sugars. And the males, since they never take a blood meal during their lifetime, only feed on nectar. And since these mosquitoes are collecting nectar, skipping between flowers, they have the potential to pick up pollen along the way and spread that around. So a role as pollinators in the environment as well. So yeah, pretty important and interesting ecological role with mosquitoes, along with the public health role as well. So now looking at the climate and environment of BC and the Yukon. So as you can see from these figures here, there's quite a wide range of ecozones throughout these regions. And that can range from wetlands and boreal forests to cooler alpine areas, drier prairies, and also coastal marine environments as well. And with this variety, not just in climate, but also in vegetation communities, it does mean that there's lots of different habitat for different types of mosquitoes to exploit. And in that, being able to showcase some different adaptations that they have to use and the life histories that they've adopted in order to survive there. So now onto the mosquito diversity of BC and the Yukon. Currently, there are more than 50 species present in BC and more than 30 in the Yukon as well. Most of these are in the genus Aedes, but we also have several species of Anopheles, Culex, Culicida, and Coquilitidia as well. And these genera have adopted a lot of individual characteristics that can reflect some of the habitats that they use and their life strategies. So for Aedes, as an example, they tend to lay their eggs in more temporary water areas. So for example, tree holes that collect water, they can lay their eggs there, or in more urban areas, if there's discarded containers, that's a good spot for them to lay their eggs too. As for overwintering, they tend to do that in the egg stage, but some species are also known to do it as larvae. And this is in comparison to Anopheles, Culex, and Culicida. So in contrast, they tend to lay their eggs in more permanent bodies of water. And instead of overwintering in the egg stage, they overwinter as adults. So as I mentioned, going into that sort of hibernation, coming out when it becomes warm, and they lay multiple rounds of egg laying during the warm cycle per year, which is pretty cool. And finally, Coquilitidia, which are pretty neat. So Coquilitidia tend to lay their eggs near emergent vegetation. And the reason that they do this is actually because their larval stage is a little bit different from the typical mosquito species. So normally, mosquito larvae have a siphon at the posterior of their body that they angle up towards the top of the water, and that's how they get their air and oxygen. But Coquilitidia actually have a more sclerotized siphon, as you can see on my little left picture here, that they can use to pierce the roots of these emergent vegetation, such as cattails, for example. And then they derive their oxygen that way, which is pretty cool. And this is also important because they overwinter in their larval stage. So if there's an ice cover or something preventing them from reaching the surface, they can still get their oxygen from the surrounding vegetation. Yeah, another cool thing about them, they have a pretty long flight range, which also has implications for disease control as well. But yeah, so most of these species in BC and the Yukon are native, but there are a couple that are invasive. For example, this picture on the right here is a picture of Aedes japonicus mosquito, which is invasive to BC. And it's sort of recognized by its darker body color with those beautiful little bronze scales and sort of a lyre shape on the back of its thorax and the scutellum. So that's just one example of an invasive species. And with that, I will pass it off to Dan.
Great job, Danica. Thanks for setting us off on the right foot there. So of course, why we're interested in mosquitoes in particular is because they are some of the world's deadliest organisms. This is a subset of species that are found in BC, both native and invasive, and you can see all the things that they can vector there. And so just to run through these really quickly, we have Aedes japonicus, which Danica just talked about. It's found in BC. It's invasive. In its native range in East Asia, it's a vector of Japanese encephalitis virus, which is asterisk there. The asterisks here indicate that pathogen isn't yet present in BC. Whereas bold pathogens indicates that not only is that something that at least at one point has been here, but the species has been implicated as a primary vector in a field setting. So Aedes urincus, the western treehole mosquito, is a vector of dog heartworm, and it vectors that here in BC. We have Aedes toboi, which we don't know whether that's a native or an invasive species. I'll talk a bit more about that one later. But in East Asia, where it's also present, it's a vector of Japanese encephalitis virus and Brugia malayi. We have our Anopheles earlii, which is found all the way to the Arctic coast. Anopheles freeborni, the western malaria mosquito. There were possibly some historic endemic malaria cases in southern BC. There's not a lot of information on them, but there are some old publications from the late 19th and early 20th century saying, hey, if there's malaria here, somebody should come look at it. And as far as I can tell, it was never followed up on. But in the vicinity of Karameas, anyways, it looks like there was malaria concurrent with malaria outbreaks in Washington state. And it's thought that Anopheles freeborni was the primary vector. We have Anopheles punctopenis, which is also a competent malaria vector. Our main Culex species, Culex pipiens, Culex restuans, and Culex tarsalis. It's a bit misleading to call Culex restuans a main Culex species. This is something that was found once in the early part of the 20th century in BC, and there was uncertainty about whether or not it was actually here, or whether that was a misidentification, or it was a one-off that kind of came in on a boat or something and then died off. But just a couple of years ago, a colleague of mine, Sean McCann, who's an avid natural history photographer, was taking pictures of all sorts of stuff in Goldstream Provincial Park. And one of the pictures he took was of a mosquito. And we looked at it a couple of weeks later, it was this species. So this might be here and is just rare, but we don't really have the survey intensity that we would need to call that either way. I'll talk about that a bit more later. But either way, these three species can all vector West Nile virus. Culex tarsalis can also vector Western equine encephalitis, possibly vector Japanese encephalitis virus, and a whole host of other things. I've had this described to me as being the mother of all vectors. It was traditionally thought to only be present in southern BC, but a couple of years ago I found it up in the Yukon. So there's some interesting stuff possibly going on there as well. And then the Cochleatidia perturbans that Danica mentioned there earlier. These guys can vector West Nile virus. They also can vector Eastern equine encephalitis virus, which isn't in BC, as well as stuff in the California serogroup viruses, such as snowshoe hare virus, lacrosse virus, and Jamestown Canyon virus. These are sort of understudied, probably underdiagnosed, and we don't really have a great handle on where they are and what they do. Although snowshoe hare virus does seem to be present all over BC and the Yukon, and possibly some of the other ones as well, but there's a lot of unknowns with those. But I'll focus on just the West Nile and Western equine encephalitis for just a second here. These tend to be present mostly in southern BC and in the interior. And this is likely going to be where most things are going to be the highest intensity if we do get, emerging diseases show up here, just because this is where it's the warmest in the province. But as climate changes, these things might spread. So while they're somewhat limited in their distribution currently, that might not be the case in the future. So we have here just a couple of examples of climate attributes that are expected to change in the future. And these are thought to be the two that are most important for mosquitoes. So temperature and moisture, precipitation and humidity are thought to be the factors that are most important in determining mosquito distribution. So temperature obviously will affect what can survive where, and that looks like it's going to change a whole bunch, particularly up north. The farther north you go, the more temperature is expected to change, and that'll interact with moisture. So you get different precipitation patterns, you'll get different species that can breed in different areas, and you'll also have species surviving for longer periods of time when it's more humid out, because desiccation is a high source of mortality for adult mosquitoes. So this is a rough idea of what things are going to look like in the later half of the 20th century, or 21st century, I should say. So yeah, things are going to warm and precipitation patterns are going to change, and therefore we expect mosquito distributions to change as well. And obviously that's important because without the vector, the pathogen can't be transmitted. If you have the vector there, well, then the door is open for the pathogen, or if a new pathogen shows up, you sort of know where it can spread if you know where the vector is present. But thinking about climate change, so I mentioned that temperature and precipitation are the key factors, but it's not just the mean averages that are important. It's also how they're distributed. So if you get spikes in temperature in the winter or the summer, versus if it's just warmer, the same temperature for longer, or if it's both, those will also affect the different types of mosquitoes you'll get. So for instance, warmer, shorter winters might make it easier for some of these overwintering species, the species that overwinter as adults, to survive in places that they otherwise wouldn't. Same with those that survive as larvae. They can survive in water under ice, but if it freezes completely to the bottom, then they'll die off. So it's with them, it would be sort of a threshold effect combined with a duration effect that'll determine the depth of that freezing. So it's not just the average temperature, but it's the pattern that's going to matter as well. Also, warmer summers. Well, if it's warmer for longer, species like Anopheles and Culex and Culecida should be able to build up their populations quicker, but also to higher levels because it's warmer for longer. And this will also affect pathogen transmission rates because you'll hit those intrinsic incubation thresholds quicker or with more consistency. Changes in precipitation might affect change distribution in species like Aedes, where the eggs are laid in temporary sources of water. In addition to the tree holes and the containers that Danica mentioned, these are also species that will breed in meltwater and floodwater pools. So when you get water levels build up or snow melt, they'll use those ephemeral sources of water. And if you get, repeated flooding events or repeated heavy rain events, you can get repeated generations of these things building up throughout the year. We also have to consider shifts in mosquito community ecology, because mosquito species compete with each other in the larval stage. They'll compete for resources. So you might get species pushing each other out of habitats. So you might see non-vector species being replaced by vector species, even though it's the same niche. Changes in temperatures might just allow the situation to be competitively advantageous to one group over the other. You also might see this with invasive species being able to outcompete native species in areas that they weren't able to do that previously. So it's not just the vector species that we need to think about. We need to think about mosquito ecology as a whole. And we also need to realize that we might start seeing invasive species show up that weren't here previously. Just things that cold or nature might have taken care of previously. If you get just one or two individuals show up, and then you get a cold snap, kills them off, no problem before they can establish. Well, that might not be the case anymore. We also might see changes in the movement of animal hosts that could affect what pathogens or reservoirs are available out there in the environment for a mosquito to encounter. So again, it's not just the vector species, although it is that too, but we have to think of a whole bunch of different things, and that is sort of compounded by knowledge gaps that we have currently. And these are things as we have lots of knowledge gaps, and some of them are as fundamental as what species are here, where are they, and are their distributions changing, or will they change? And this is pretty fundamental stuff, but that's how far our ignorance in BC and the Yukon goes. I mean, in the Yukon, including myself, there have only ever been 5 sort of major collecting efforts that have been at least put out there, and that people are well aware of. So there have been more, I'm sure, of people going through on their weekend kind of thing, or maybe stuff that isn't out there and has been published. But there just hasn't been a lot of survey attention paid to a lot of places outside of the lower mainland. So we really don't know a lot of what's going on, and we might not be able to answer parts of the second question about will distributions change, or are they changing? Because we don't know if we find something new somewhere. Was that there all along? Was that not there previously and it just showed up, and that's why I found it there? If nobody's been there looking for stuff yet, we just won't be able to know. Now, there are things that have been done to try and combat these knowledge gaps, such as surveys and trying to tie in citizen science. Excuse me. But there are also challenges from these. iNaturalist is a fantastic program that allows for biodiversity surveying for a lot of organisms, but it tends to be organisms that are big and easy to identify with the naked eye. For mosquitoes, I mean, even if you have a pretty skookum camera lens, a member of the public that's out there taking pictures might not know exactly where to point that lens to pick up the cues that you need to be able to identify a species. There are also, people will go walking where it's nice to walk, or where they happen to be anyways. People aren't going to go plunging into the middle of godforsaken mosquito-infested muskeg just to take pictures and upload for people. So there are some limitations for this, and that's also assuming that the mosquitoes are in good shape. So mosquitoes have scales on their bodies like moths and butterflies, and the patterns and distributions and colors of these scales are often useful ID characteristics. But if they've been bashed around at all, they'll often fall off, and you won't be able to tell what species it is. Now, to some degree, you can use molecular techniques to address those shortfalls, and I'll talk a bit more about that later. But there are problems there as well, where you have to assume the species has been properly identified first, which isn't always the case. So you could be comparing against a reference barcode of something that isn't actually from the species that is reported to be from. You'll also get, there are cryptic species complexes out there, so you might not know if what you're comparing against is actually the same species that you think you're looking for. And there are undescribed species out there, so there just might not be references for what you're looking for. Now you can use specialized mosquito surveys to overcome these shortcomings, but they really have to be specialized for mosquitoes. Even a general entomologist out there collecting doesn't tend to get a lot of mosquito specimens that are in great shape. So if you think of somebody out there with a butterfly net, well, that net's going to rub off scales and rip off legs. Or if they catch a mosquito and put it in a jar of ethanol, that's gonna disfigure it and make things more difficult. So there really is a need for specialists who are trained in mosquito ID or trained in special mosquito activities to go out there and look for mosquitoes or set up mosquito collecting traps, whether that's a scientist or a public health official or a mosquito mosquito professional of some sort. So our current work is trying to address a lot of these knowledge gaps and then figure out solutions going forward, or at least project what we can with what information we have going forward to figure out where are these species, where are they going to be in the future, and correspondingly what pathogens might show up that could spread with these species, or could be vectored by these species, or how might some of the existing pathogens change in their distribution. So we're doing that in a few different ways. One is using surveys. So that's whether it's incidental stuff, and I'll talk about that in our next slide, or going out and doing formal mosquito surveys, which I spent some of my postdoc time at UBC doing. I don't have all of the data processed from all of these yet, so a lot of it's still work in progress. But we are going to be creating habitat models using some machine learning techniques to figure out, okay, we have some data on where stuff is. We can use these as a force multiplier. You still need that base data, but you can use modeling techniques as a force multiplier on that to get a more extensive view based on confirmed occurrence records. And we are also tapping into citizen science as a sort of supplementary method to boost our data. Again, it's no replacement for the fundamental surveying activity, but it can help supplement that. And so I'll go through those as we go through this presentation, but that's what we're working on here. And it's important to consider that ideally we want lots of information about where the mosquitoes are that overlap with where people are. But again, there's stuff that goes on with animals and out in the environment that doesn't directly involve people, and so it's good to know what's going out in the periphery as well. And so, for instance, in this picture here, I'm up in some alpine habitat, 1 or 2 valleys over from Whistler, looking at what's going on there, because you could imagine that if you were to start seeing pathogen transmission occurring up in Whistler, they'd be pretty upset about that due to the lost tourist revenue, let alone the health concerns. So these are some range extensions that I've come across just with some incidental surveying over the last few years. So these are not targeted methodological things. They are from, for instance, the Aedes spencerii and Aedes sticticus up there, which are both the first records of those species for the Yukon. That was on a camping trip with my wife. Cochleartidia perturbans. Our first record there was meeting up with a friend outside of a bar in Watson Lake, and he just happened to have this species land on him. I said, hey, that's not supposed to be here, or it's not known to be here. Let me grab that. And sure enough, it was a new species record for the territory. So that speaks to just both how easy it is to pick up some of these new records, like how low the bar is for we just need to put in some sort of effort, to the need for having a specialist involved, because it lets you pick up on stuff that you wouldn't otherwise do so. Real quick, we have Aedes uides and Culex tarsalis found in the Yukon that weren't previously known to be there. Aedes decticus, that's just over the border in northern BC, but it probably comes much farther south. It's a bog mosquito, and not many people go tromping through bogs looking for stuff. Our Culex pipiens, that's a northern range extension. I'll get to that shortly. And then we have Aedes nevadensis. It was known to be here since the 80s, but there weren't formal records of it. So these are the first formal records of that species, including northern, as far as the northern limits of their records. So Culex tarsalis, the western encephalitis mosquito, it's a native species, but we never really had a great handle on how far north its range extended. And so you can see in this map here, we have a whole bunch of records in southern BC. They're in the southern prairies and stuff too. But in the 1970s, there was a record from the Northwest Territories. You'll see up there, and that's in Norman Wells. And nobody really knew whether that was a one-off that came up in somebody's car who was driving up to a work camp from Alberta, and it just kind of hitched a ride and died off at the end of the summer and was never there again, or if it was a recurring thing. Well, then a couple of years ago, we were at a bioblitz in the Yukon Territory, and we stopped off at one pond near Slims River's Flats and found some larvae of this thing. So it was not only there, but it was breeding. So it certainly seems that it's there now, but again, we don't know if these are range extensions, like this species have been moving north with climate change, or if these were here all along and just went undetected. There was also a recent record from Alaska. So it's definitely north now, but was it there already? Or if not, how long has it been there? We don't know. What about in between? So we're hoping to be able to pick up from some of these citizen science projects that I'll talk about later, whether or not this species exists in between these points. Hopefully we can fill in that knowledge gap, but it's an unknown at present. Culex pipiens, the common house mosquito. So this is an invasive species. It's been here over a century. It was first recorded in the early 20th century in and around Vancouver and Victoria. It was quite rare at that time, but it's spread around since then. And is it still spreading, or has its range been shifting? It's a bit of an open question. So this is a map of Canada here. And these dashed lines represent the accepted limits of the distribution of Culex pipiens in the last sort of continent-wide guide for the distribution of this species, although it's a bit out of date now. You can certainly see that this has been found north of that in quite a few places, both in BC here, in Prince George, and in southeastern BC, as well as in the Maritimes. And then there's this one in Quebec, this one record there. So we don't really know if that is an established population up there, or if that is a one-off. And I'm told that there are also records in other parts of Ontario as well. But I haven't been able to get some specific information on that. And there's lots of great folks in Ontario doing work on that. So I'll leave that to their purview. But what's sort of interesting here is that this, in the 1890s, Culex pipiens was detected in Sitka, Alaska. And whether or not it had occupied these northern ranges and just wasn't there at high enough levels to be detected, or whether these are recent expansions is sort of a question mark as well. Aedes togoi, the Rockpool coastal Rockpool mosquito. This one is particularly interesting and really highlights how little we know about what's going on with mosquitoes in BC and the Yukon. So this, we're not even sure if it's native or invasive here in BC. It was originally described in Asia in 1909. A specimen collected in Victoria was found undated in the Canadian National Collection in 1970. This was from somebody's personal specimens that were donated when they passed away in the late 1960s. Now, this person moved to Victoria in the early 1940s, and based on that and where this specimen was found in their collection, like the dates on the things around it, it could have been collected as early as the 1940s. But as soon as this was sort of on people's radar, they went out and looked for it and found it all over the place, up and down the Georgia Strait and Puget Sound. It occupies these, they're called supralittoral tide pools, and they tend to be in rocky headlands. So think Lighthouse Park in West Vancouver kind of deal. So it's not really a typical place you'd expect to find a mosquito. And also when you get close to these pools, these things are very skittish. So they'll dive right to the bottom, and they tend to stay there longer than most other species would. So they're easy to disturb. They're not in a place people would typically go looking for mosquitoes. And they're sort of a weird thing, but because of how they were originally found here, there's sort of this question mark. Were they here all along? Like come across the Beringian land bridge in the last ice age, or did they come across in shipping from East Asia at some point? And it's sort of an interesting question. We have done some work to try and answer this, both myself and some other folks. Interestingly, our population here is not genetically related to any known population in Asia, except for some specimens that I was able to get out of the Russian Far East just a couple of years ago, before the recent stuff with Russia has started back up again. So it does seem that their stuff we have in BC is related to stuff from the Russian Far East, but whether that's due to an introduction, either through, you know, the Russian sealers and fur trappers that came down here, or shipping, or the Canadian troops that were sent over to fight the Bolsheviks after the First World War, who knows? But they're here and they're closely related to what's in Russia, but whether they're invasive or natural is sort of unknown. But that's just how deep our lack of understanding is. We don't even know how far up the coast these things go. And this is some work we did, though, with a habitat modeling to try and elucidate that. So this is done with MaxEnt, which is maximum entropy niche modeling. We basically took our records from North America and tried to figure out what other habitat was similar to the habitat they currently exist in. This is what it looks like under current conditions. So it seems like if they're native and if our sampling gives a decent representation of all the places in North America that they're found, well, this is where their current extent of their range is going to be. If they're present elsewhere, well, that would change these, but these are our tentative results. Now, interestingly, we can project this forward in time to see how these distributions might change. So it looks like going forward to the year 2050 under moderate climate change scenarios, and the lighter the color, the better here, you'll get better habitat for these critters, and you might even start seeing them up on the north coast. So if we want to know whether this was present naturally, we should probably go up there and look for this thing before it gets too late and we start getting good conditions up there based on what we know about the population we've already sampled. We've also had Aedes japonicus come through lately. This is an invasive mosquito first arrived in North America in 1998, Northeast USA. Was first reported in BC in 2015 from 2 locations in the Fraser Valley. These are distribution records for it across North America. I've since found it in 2 or 3 other places in the Fraser Valley, as well as a record from southern Vancouver Island. But I took these records and put them into our machine learning program. And this is sort of what we were able to get for suitable habitat for this critter across North America under current climate conditions. So it looks like it's still spreading and will continue to spread in both the Pacific Northwest as well as in the US. So it probably will turn up in Louisiana and Texas at some point. We might see it go up the north coast of BC and down the west coast into maybe even Northern California. This is just the same map in a different format. So we took this and projected it into the future. And it looks like, again, the lighter colors, the better the habitat. It looks like we will start to see northern shifts in this species. So it would probably be a good idea to monitor for it and figure out where it's going to, you know, as it moves, where are sort of the front lines of its distribution. So this is another thing that we're hoping that we'll get some citizen science picking up. But yeah, I'm getting a bit ahead of myself there. Well, only by one slide, it turns out. So we've been doing some work lately. This was at UBC that now that I've gone and started up my own lab, I'm still going to be involved in this project remotely. Danica is being involved. She's been working on it for quite a while now. She's a very, very talented student with a bright future. But we're going to work on barcoding every species that we can get our hands on from British Columbia. This is both to make sure that we have a voucher specimen that we are confident in what it is, that we have a barcode associated with, and also to make sure that we have this material from our sample areas of BC and the Yukon, so that we're not relying on specimens from elsewhere where there might be greater likelihood of cryptic species complexes or different genetic diversity that might confound things. So it just sort of covers all of our bases here. It also allows us to sequence some novel barcodes. So we have a few species so far that we've got novel ITS2 barcodes for, things that don't have these barcodes out there yet. And we're combining this with a citizen science project called What Just Bit Me. And this is something we're very excited about. We've been on a sort of media campaign the last few months leading up to the summer to get this started. And so what we've been pitching to the public is, you know, hey, everyone's familiar with mosquitoes, right? We smack them, but you never really know what that is that you just smacked. So why don't you mail it to us with the location of where you got it, put your name on there, the date, and send it to us, and we'll barcode it, we'll figure out what it is, and then, hey, we can let you know what that was, and then we can use that data as survey data to figure out, okay, this species was present at this location on this date, and we can use that in our habitat distribution models to try and plug in some of our gaps. Now, there are some flaws in this, and that it will certainly be biased towards where people are. We've been trying to account for that by reaching out to outdoors groups, hikers, guiding outfits, First Nations communities, just wherever we can, to get people from places, outside of the southern Vancouver Island and the lower mainland and South Okanagan, to get them involved in this too, because they're going to be increasingly exposed to mosquito-borne pathogens, and they're some of the areas that we know the least about. So we're hoping to be able to use this to make models like the ones I just showed for as many species as we can for British Columbia and the Yukon. At some point, I'd love to do this for all of Canada, if not all of North America, but we need much more data for that. So that's what we're doing, but some things to keep in mind, going into the future, why we're doing this, why it's important, and also things that we're not able to do with this project, but that we want to put out there as ideas and maybe spur either discussion or just put it on people's radar if they have the ability to make these things happen, to at least discuss them. In the future, it's going to be increasingly important to monitor for mosquitoes at airports and seaports, to the extent that we're able to do, and that there's money to do so, or the political will to do so, or what have you. But mosquitoes absolutely hop around on planes, as well as on ships. I mean, the reason that mosquitoes first showed up in Hawaii was due to being presence in some bilge water, ballast water on a wooden sailing ship, and to this day, mosquitoes still show up in new areas based on on being in shipping holds or in containers in ships, as well as in airplanes, whether it's the cargo bay or what have you. So on the right hand side here, we have some data that came out of China in 2019, where they were monitoring the invasive mosquitoes that were showing up at airports and seaports, a selection of them anyways, in Zhejiang, I hope I'm pronouncing that right, province. It's a bit murky on how much survey effort was done, and it certainly wasn't every vessel or every airplane that came in, but whatever subset that they did monitor, there seems to be an increasing trend, both in the diversity of species that were showing up, as well as the number of invasive species that were showing up. So these things likely will be showing, we will start getting these things here if we aren't already. And as our climate changes, some of them might start taking hold, whereas they wouldn't have been able to do in the past. So, I mean, this could even be as simple as, hey, maybe we should have a permanent, mosquito trapping system in place around YVR in the Port of Vancouver. That something like that might be a simple enough solution to see what's coming in. But we also have a few other things going. So there was a study that came out of Spain not long ago that showed as many as 5 out of every 1,000 cars in Barcelona had an invasive Aedes albopictus mosquito in it. So invasive species that show up in one part of BC or the Yukon or one part of Canada could very well just hop around domestically in people's cars. And so this is it. Really gets the message in. We need to do some monitoring for this. A lot of our mosquito control organizations here in BC will just, they're basically pesticide delivery mechanisms. They don't always identify the species that they're treating, and when they do, they certainly don't make the data public. There are some that if you reach out to, they're willing to do that and share it with you, but not all. I know other provinces, for instance, Ontario, Public Health Ontario has at least to my knowledge, done a pretty good job of monitoring for mosquito species in Ontario. So I'd love if we could get some sort of monitoring activity going consistently in BC and sharing the data so that it is accessible for people that need it. Again, Ontario is great for this. I've reached out to them a few times to ask for this, and it's fantastic. But we don't really have a centralized way to do that here. And we also need to think about mosquitoes, one health context going into the future. So it's not just the mosquito and the person that they bite that matters. There's animals out there, as well as the environment. So and a prime example of this would be, poorer neighborhoods tend to have more litter, and litter, if there's stuff that can hold water, that can create breeding habitat for invasive mosquitoes. Or agricultural activity like development of rice fields. So that's great habitat for all sorts of vector mosquitoes. So keeping these things in mind as we go forward and as these issues arise is very, very important. And I'm just going to go through a few examples of invasive stuff that should probably be on our radar as I try to close out here. So Culex stigmatosoma is a species that's native to the U.S. It breeds in areas of high organic water content or high organic content in water, such as winery waste, sewage, cattle waste ponds, log ponds. Sounds a lot like stuff that's present in the Okanagan, right? So this is a species it's scattered distributions in Washington. If it's not in BC native and already, it probably will be soon. It's not that far away, and as things warm up, this could certainly move farther north. It's a West Nile vector. It doesn't bite people very often, but it's really good at ramping up West Nile and bird populations. Aedes albopictus and Aedes aegypti. Well, Aedes albopictus was recently found to have established in southern Ontario. Aedes aegypti tends to be collected now and then there as well. So it's not impossible that one of these could hitch a ride west in a car. Could also turn up on its own. For instance, Aedes albopictus showed up in some imported tires in Seattle in the 1980s. And luckily, this was detected very quickly and a very aggressive response was taken to eradicate it. But had it not been picked up quickly, it might have spread far enough that it wouldn't have been able to be eradicated. So this is really a good case study in why it's important to monitor for stuff here in BC. You can see on the right-hand side, there's some modeling data showing that, pretty soon there's going to be suitable habitat for, if not just Aedes albopictus, both Aedes albopictus and Aedes aegypti in southwestern British Columbia. Depending on which data you're looking at, there might already be suitable habitat for Aedes albopictus here. So this is not some far off thing. This might be just around the corner for us. That it's something that we need to have on our radar now. Another possibility is Culex Trine urincus. So this is the primary vector of Japanese encephalitis virus in the field in East Asia. It's native to Southeast Asia, but it's been spreading quite a bit lately. It's found its way all the way to Africa and to Europe. It goes up into Japan and into Russia. So could possibly establish here in BC were it to get here. And this breeds in, it's a container breeder. It also breeds in rice fields. So it can be moved around in containers. We're starting to get a few rice fields established around the lower mainland in British Columbia. So stuff like this, if it were to get here, could just hop right into habitat that it's already known to use. And adults also, for whatever reason, turn up on ships hundreds of kilometers offshore. And it's thought that they might get up into the air column and then see lights and fly down to them and land. I'm sure to some degree it's also stowaways that are these adults of this species will hunker down in these ships and then they're found. So it's spreading. It could show up through shipping at some point. I believe it's not shown on this map, but I believe it was recently found in Australia as well. Whoa. Let me get back to where I was there. So Japanese encephalitis virus is a pathogen that's been on several organizations' radar for a while as an emerging pathogen. It's endemic to East Asia, including areas not too dissimilar from here, like parts of the Russian Far East and northern Japan. There are rare imported cases in Canada, but so it does get here. All it would take would be a vector like Culex tarsalis to bite somebody that has it and then transmit it into the environment, and it could establish. The animal reservoirs for this are pigs and wild birds, including some groups of wild birds that end up migrating from Russia to North America. Sometimes it's accidental migration. Sometimes it's just rare events that they do every few years. But for instance, things like herons can be reservoirs for this. And so as it's not just the effect of climate change here that we need to be concerned about, but as things warm up in East Asia, you might get more and more infected birds farther and farther north, increasing the likelihood of infected birds accidentally ending up in North America, and bang, you have the pathogen introduced. So I'm just going to wrap up really quickly here. If you're interested in reference material on mosquitoes, there's a few great books out there, including the Mosquitoes of British Columbia by my late mentor, Peter Belton. It's unfortunately about 40 years out of date now, so I'm working on an updated copy. It'll be out at some point, though it's probably been delayed a bit by my new job, but it is still coming down the pipeline. We've got a guide to the mosquitoes of the Yukon that came out a couple of years ago. And then we have broader guides. We have the identification and geographic distribution of the mosquitoes of North America, north of Mexico, which is it's more than a decade old now, but it's very, very good for mosquito ID as well as broad mosquito distributions, although its information on Canada is sort of hit or miss, and for BC and the Yukon in particular. We have the Mosquitoes of Canada, it was published in 1979. It's quite out of date, but it is really, really good for the information that was available at the time. It's a great baseline. And then if you're just looking to identify adult mosquitoes, there's a really great photographic key to the adult mosquitoes of Canada that's available on the in the Canadian Journal of Arthropod Identification, which the Biological Survey of Canada puts out there, and it's open access. Anyways, I'd like to thank a few folks here. My late mentor, Peter Belton, who's the one who sort of mentored me in mosquito ID and getting me involved in a lot of mosquito natural history. My PhD supervisor, Gerhard Gries, who taught me a whole lot about ecology and chemical ecology, and just great ways to tie natural history into more technical or mechanistic publications, as well as just being a great general mentor to me. My postdoc supervisor, Dr. Ben Matthews, Karen Needham at the BD Biodiversity Museum, who's been involved in countless collecting excursions with me, as well as getting me involved in mosquito identification, and Joshua Chen, who's an undergrad who's been working on doing a lot of the barcoding with us, as well as it's a project Danica is going to be getting more and more involved with here as she finishes up her undergrad degree. Also funding from the Banting Postdoctoral Foundation, the Michael Smith Foundation for Health Research, Yukon Conservation Data Center, the University of British Columbia, the BD Biodiversity Museum, and the Whistler Naturalists. So I'd like to thank all these folks, and I'll turn you over for questions just a sec. But Danica, I don't know if you want to have a sign off here too.
Sure. Yeah. Thank you so much for having us. It's a really cool opportunity to get to talk about some neat mosquito stuff. And just as a slight shameless plug, I'm going to be moving on to my master's hopefully after my undergrad. So if you happen to have any positions opening up in your labs in September 2023, I will put my email in the chat. Thank you so much.
All right, we'll open up for questions.